Guys, welcome back to my channel Zoologist AK, and I am Aarti Kaushik again here with another video. And in this video we are going to discuss about principle of inheritance and variations. Here I am using the word inheritance. Inheritance means passing on of traits from parents to their offspring. Like my daughter or son will look like somewhat similar to me, like it could have happened that his or her hair will look like just look like I have the type of hair, and it could also happen that my iris color will match to my daughter iris color. So these are some example of passing on of traits from parents to their offspring. It could have happened either through asexual reproduction or sexual reproduction. The offspring cells, or we can say the organism, acquire the genetic information. I mean to say how he or she gonna react to the atmospheric condition, either the genetic variation he or she is going to receive. Like if I have asthma, I could pass on this to my offspring. If I have any genetic mutation in my gene, like if I have a BRCA1 mutation, that is BRCA1 and 2, that result in causing cancer. If I have that gene mutant form in my genome is the haploid set of chromosome that is present in a particular cell. So there are high chance that I am gonna transfer these genes to the offspring I am going to reproduce, and that offspring is going to have cancer in future. So these are some example of inheritance of characters or traits from parents to offspring. And here I am using a word variation. Variations mean the differences means there are many individuals in a population. If I talk about human population, so we are well aware with African peoples, American people, Chinese people and Asian ones. All are different in morphology, in physiology, and somewhat anatomically. I mean to say, like African people have more twins as compared to Asian peoples. So and their height, weight, their facial expression, and their capabilities, or I can say the adaptation to the heating and cooling environment is different. So these are result into a species like Homo sapiens is a species, and these variations are result from the sexual reproduction that is happen between the organisms. So in this video we are going to understand how does the Gregor Mendel explain the principles of inheritance and variation. Means how does this inheritance occurs, and how does these variations takes place in a particular individual of a population. So stay tuned with me, and thank you so much for clicking on my video. Let's understand some terms and get to know how does this Mendel started his experiments on hybridization. Gregor Mendel is known as father of genetics. Why do we know him as a father of genetics? Because he has given very important principle of inheritance, like the principle of dominance, the principle of independent assortment, the principle of segregation, which we are going to understand in my upcoming video, because this video is going to be very lengthy if I include all these points in a particular one. Here we are going to understand some terminology that is very much required to understand the topic further. So the father of genetics, that is Gregor Mendel, proposed that inheritance, I have just talked about inheritance, means passing of traits from parents to their offspring. It is controlled by pair germinal units. According to Mendel, these pair germinal units were factors, but now we know that these were genes. Genes are present in the chromosome and they have a particular locus, and these genes are going to give the information to the embryo to express in a particular way. So these genes are heredity unit. Mendel performed hybridization experiment. What does it mean by hybridization? When we choose a species, like here Mendel chose pea plant that has the scientific name Pisum sativum. This pea plant, in this pea plant he has chosen the two traits. When we are going to take two traits and going to produce a new offspring by crossing them over, then the resulting progeny will consist the characteristics of both of the trait. So he has performed the experiment like this by crossing two different traits of a pea plant. That is why we call it that he performed hybridization experiment. Now here I have used a word genetics. Like Gregor Mendel is known as father of genetics. What does it mean by genetics? There is many branches of biology, and one is the most important branch of biology is genetics. It is a branch of biology that deals with the study of heredity and variation. Heredity means transmission of characters from parents to offspring, and variations means production of differences, morphological difference, physiological difference and anatomical difference. Here the hybridization experiment. How does this hybridization experiment work performed by Mendel? By emasculation process. Emasculation means removal of stamen. Stamen is the male part, the androecium part. Androecium means the male part. Male part means it consists of filament and anther. Anther is bilobed, and this anther is going to produce the pollen grains, and pollen grains are the male gamete. Male gametes. If anyone is going to remove this portion, this anther portion, that means that person is going to remove the pollen kit of the flower, and if the pollen kit is removed, that plant is not going to be pollinated and is not going to be fertilized. So there will be the requirement of pollen grains from another plant to fertilize the female reproductive organ. So this will result in cross pollination, because we are using here the different plant to pollinate or to fertilize the female reproductive organ. That is why we are all known as emasculation. And here, by the help of emasculation, Mendel performed his hybridization experiment by conducting the cross pollination to produce the heterozygotic condition in the offspring. So here the question arises, why does Mendel selected the pea plant for the experiment? First of all, the pea plant is herbaceous. We all are aware that there are different types of plant. Some are insectivores, which means they are partially photosynthetic, but their nutrition, many plants require the nitrogenous rich nutrition to grow well in a particular environment. The same way insectivores plant does. For example, Venus flytrap. It traps the insect that sit on it and get the nutrition from those insects. And there are some parasites as well, like Cuscuta. So this pea plant is herbaceous and going to produce its food by photosynthesis. An annual plant, and it can be grown 2 or 3 times in a year. So lifespan is very less, so we can study our progeny in a well framed manner. Peas flowers are bisexual. Bisexual means both the male part, that is the stamen part that is going to produce the pollen grain, and the female part that is known as carpel and sometimes androecium that is going to produce the ovule. Both are produced on a single flower. So here the Mendel could perform the experiment by hybridization experiment by removing the stamen, or by removing this male part, and this could be done by the help of emasculation, so that the cross pollination can be applied in pea flowers to generate the hybrid progeny. It can easily grow in garden, and many contrasting characters are found in pea plant. Mendel has selected 7 pair of contrasting traits of pea plant. These are like for plant height, he has like shape of pod, position of flower pod, and color of flower, color of seed coat, cotyledon coat, like there is 2 cotyledon. So the how does the seed show the color of cotyledon? Either it's yellowish green or it's pure green. Pod color, seed shape, and he has shown the allele with the capital letter and the small letter. Like for dominant trait he has shown the capital letter, and for recessive trait he has shown the small letter. And Mendel studied the inheritance of character till F3 generation. F3 generation means filial 3 generation, means for example these are parents and they are going to produce the F1 progeny by crossing between them, then F1 progeny will again cross between them and produce F2 generation, then F2 generation is going to cross between themselves and going to produce F3 generation. So this F3 generation, he has studies his experiment or his hybridization result up to F3 generation. And here we can study the number of chromosome. For plant height there are 4, and shape of pod there are 4, position of flower 4, and color of flower and color of seed coat 1, and cotyledon color 1, and pod color 5, and seed shape are 7. So there are different number of chromosomes that are going to consist the gene, and that is going to give the information for showing or for demonstrating that particular phenotypic characteristic. Now before reaching to the point of experiment or the principles of Mendel, we are going to understand some terminologies. Then first one is the factors. Factor is a word used by Mendel, and now we are well aware with this fact that we know it as gene, which is given by Johnson. So it is a unit of inheritance because it is going to provide the information to the fetus embryo to grow and to represent the traits in future generation, passes from generation to generation and control the expression of character in a particular organism. What are alleles? Alleles, for example, I am saying here that I passed on my height to my child. So here I am talking about a character, a particular character, that is height. So that height is going to be expressed in my daughter or my son by a particular gene. For example, Mendel has taken the capital T to represent the dominant trait and the small t to represent the recessive trait. So here these capital T and capital T are 2 allele of a gene that is going to express the trait height. So for a particular phenotypic trait there is a gene, and that gene consists the 2 contrasting traits or similar traits. Like here if I am talking about TT, that is homozygous, so these are similar forms of a gene, or I can say the 2 forms of a gene, that is known as allele. So this T is allele 1 and this T is allele 2. Sometimes these are contrasting traits. If I talk about heterozygote, then this capital T is allele 1 and small t is allele 2. Next is phenotype. Phenotype means external appearance. How does I look like? My face, my complexion, my hair color, my iris color. These are physical appearance are known as phenotype. Genotype means what I consist in my genome. Genome means the haploid set of chromosome. Like we have 46 number of chromosome, then I am going to say that 23 number of chromosomes are known as the genome, because that is the haploid set of chromosome present in a particular cell. So these 23 number of chromosome is my genome, because that is going to provide the information to my particular cell. Like if my muscle cells is going to produce, then my muscle cells will express particular genes for myosin protein. So here the information is consist in my DNA, my genome, that is going to form the protein, and that protein is going to express, but that protein contain the information that were present in my DNA. So this is genotype. Homozygous means the identical allele. Like I have just talked about TT, capital T and capital T, that capital T is dominant, so it is going to be a tall plant. So here both the allele are similar, and if a gene consists a trait for height and consist two alleles, and both the alleles are similar to each other, then it is a homozygous condition. But if alleles are different, like capital A and small t, then it is a heterozygous condition. And here the dominant allele will express, because the dominant allele has a capability to suppress the expression of recessive one. Hybrid, this is an example of hybrid. If I cross between pure homozygous tall plant and pure homozygous recessive plant, then all the progeny will be hybrid, and all the progeny are going to be heterozygous, means T, capital T and small t. And this capital and small t is going to produce the tall plant, because we all are aware capital T is a dominant trait, and dominant trait is going to suppress the effect of small t. So it will produce the progeny that will have a phenotype of tall pea plant. So these are hybrid. Now we are going to understand about reciprocal cross. Reciprocal cross is a cross in which we are going to change the interchange of sexes of the parents, which are going to cross with each other and result in the formation of progeny. For example, if in F1 cross I am taking two parent, one is A1 means it is homozygous and it is female, and I am going to cross it with a homozygote male, that is A2, and it is going to produce F1 progeny, and that F1 progeny is going to produce heterozygote female with genotype A1A2 and heterozygote male with a genotype A1A2. But again, in reciprocal cross, I am going to interchange these sexes. Like this was the female here and this was the male here, but I will interchange the sexes here. Like I will take A2 as female and A1 as male. And here the reciprocal F1 progeny will result in the formation of female heterozygote with A1A2 genotype and heterozygote male with A1A2 genotype. So this is an example of reciprocal cross. Next one is back cross, and here in back cross, a cross is performed between F1 hybrid and one of its parents, and it includes test cross. For example, if I have pure tall plant means I have consist a plant that will have a genotype in homozygous condition, means its genotype will consist capital T and capital T in a homozygous state. And I will consist here the pure dwarf plant with homozygous condition small t and small t, and this is the parents, and they are going to cross between them and going to produce a hybrid in heterozygous condition with genotype capital T and small t. So this is F1 generation here. And again, if I am going to cross between this hybrid, this heterozygote means capital T and small t with one of the parent, that is the dominant parent, that is capital T and capital T. So these cross will result in the formation of progeny with capital T, capital T, capital T, small t, capital T, small t, and capital T, capital T. So this is known as the back cross, because here the heterozygote progeny is going to cross with one of the dominant parent, and it is known as the back cross. It is very useful in genetic study for isolating or separating out certain characteristic in a related group of animals or plants. I have include a word here, it include test cross. So test cross the similar thing is happened, but here the F1 hybrid and recessive parent is crossed, and it is done to know either the individual is homozygous or homozygous or heterozygous for dominant character. For example, in back cross I have talked that for I have chosen the parents with homozygous condition capital T and small t means both the parents are homozygous here, and the cross result in the formation of heterozygote with capital T and small t. So it is a hybrid here, but in back cross I am going to cross between this hybrid and the dominant parent, but in test cross I am going to cross between this hybrid with the recessive parent. So here I wanted to cross to know either the individual is homozygous or heterozygous if I am unaware with the genotype of the progeny. Let us understand it with an example of two cases. In case 1, if parent is homozygous, here, then homozygous parents, then capital T tall plant is going to cross with dwarf plant and going to produce the progeny with heterozygous phenotype or heterozygous genotype, that is capital T and small t, but all the plants will be tall, as here the progeny will consist genotype in heterozygous condition, and in heterozygous condition we all are aware that dominant allele is going to express, and dominant allele here is capital T, which is going to produce the tall phenotype. So again in case 2 we are going to cross this tall plant, that is the progeny in heterozygous condition, but we are not aware either it is a heterozygous or homozygous, because we are not aware with the genotype. We have just crossed and we have just looked at the phenotypic characteristics of the plant, and we received that plants has a characteristic of tall plant because it looked like a tall plant, but we wanted to know the genotype, so we are going to perform the cross further. So we have chosen the progeny here and going to cross it with the recessive parent, that is small t and small t. Then all the progeny are not going to be tall, because here it will produce the progeny capital T and small t, capital T and small t, and small t and small t, and small t and small t. So 50% progeny will be dwarf and 50% progeny will be tall. So it will tell us that the progeny that is produced in F1 generation was heterozygous. So test cross is performed to know the genotype of the F1 progeny, either it was the heterozygous or the homozygous. Next is genome, which I am talking again complete set of chromosome, actually haploid number of chromosome present in a cell of an organism. Gene pool, sum of all the gene and their allele present in an interbreeding population. Interbreeding population is a species, because in a species any individual can mate and copulate with another organism and going to reproduce the fertile offspring. That is why we are aware that species are interbreeding population. But if two species are there like lion and tiger are there, they are going to reproduce, but they are not going to produce the fertile offspring, they will produce the sterile offspring, so they are different species. So gene pool are the total number of genes present in a particular species. Phenocopy, when the different genotype produce the same phenocopy due to different environment. means different genotype, either it is capital T and small t or capital T capital T, both are going to produce the same genotype, same phenotype, not genotype. Genotype will be different but phenotype will be same due to different environment. But here the effect will be due to environmental condition only, that is known as the phenocopy, because it is result in the production of physical appearance, physical characteristic due to the environmental condition. That is why we all known it as a phenocopy. Pure line, it is a strain of genetically pure true breeding individuals like capital D, small t, these are pure lines because which have devised by single self fertilized homozygous ancestor and identical homozygous ancestors.